I'm Rodrigo Fernandez Gonzalez. I'm an associate professor in IBBME, the Institute of Biomaterials and Biomedical Engineering, and we are in the Mars building. So I'm cross-appointed to Cell and Systems Biology, and we're in the Mars building because I'm also appointed a member of the Translational Biology and Engineering program of the Ted Rogers Center for Heart Research. How do embryos repair damage? And the reason we're interested in that is that embryos are very good at healing wounds. So an embryo can repair a wound not only fast, but also without scarring. And of course, scarring is one of the main features of wound healing in adults, in humans. And scars limit tissue functionality. So if we can understand how an embryo is able to repair a wound without forming a scar, then maybe we can enforce humans or drive humans to those modes of wound healing, which we know humans can also do because human embryos and babies repair wounds also without scarring. So that's what we're trying to understand. Also, this process of embryonic wound repair mainly involves cell movements, and that's why I was referring to with cellular behaviors, very similar to what happens in metastasis. So there's also the hope that by understanding how cells move in a wound in an embryo, we may understand how metastatic cells spread cancer. My understanding is that it's the efficiency, right, of the transformation of the cells and how many cells you need to try to transform to get effective transformation and effective editing of the genome of those cells. So, and we see that. So in my lab, we use CRISPR for so we work with fruit fly embryos, and the fly embryo, the fly in general, is well known for being a genetically tractable system. So we use CRISPR to introduce mutations in the genome, but the efficiency of the process is very, very low. So you need to do it with many animals until you get one where the editing has actually happened where you want it to happen and how you want it to happen. So if I think about it, right, I mean, I have 2 children, and my personal opinion is that I would not want my children to be sick. So personally, I think that gene editing, when you can avoid disease, for me is morally acceptable, ethically acceptable. If you tell me, well, gene editing to, I don't know, create babies that have specific eye colors or that do grow hair, or I think those applications I can find reasons to object, because what if now we all want to be blue-eyed, right? And then we're going to run out of people with darker eyes. Well, that's I don't see the point of that. So gene editing for aesthetic purposes, in a way, I don't find justifiable. If it's to prevent disease, I don't see reasons to object to it. That's actually an example that that is very pertinent, right? Because if you ask people who have children with these conditions, right, or for instance with Down syndrome, they will tell you that those children are gifts, right, for them, and that they've learned a lot from those children. So who gives me or anyone the right to say, well, we need to prevent Down syndrome, right? And this is where it's very difficult, right? Now, I can ask the question, and maybe this is very politically incorrect, but I can ask the question, would you want your child to have Down syndrome if you were to choose? I don't know. I mean, I cannot say I don't know, right? My answer would be no. But if I have a child, if I know my child is going to have Down syndrome, would I not want that child, or would I not love that child? And the answer is no. Also, right? I would love that child as much. So I think it's a very gray area, and it's very difficult. So I don't know who defines the limits, and I think every person defines the limits. The problem is when it's the researcher defining the limits for everyone, right? And I think some community, and I don't know who that community is, would need to come to an agreement of what's acceptable and what's not. And not everyone's going to be happy with that decision, but that's society, right? The first thing, I think, as scientists, we have a moral duty to talk to people about what we do, because they are funding what we do, right? I mean, we have public funding. We get money from CHR, from NSERC, from public sources. So there should be venues where we could communicate our results to regular citizens, right? The problem is that we don't promote that. The funding agencies don't promote that either. So maybe a clause when you receive funding should be that you're going to give this many public lectures on your work in a bar or in a theater rather than in a lecture hall at a university. That's not enforced. So I can say that the Ontario government has early researcher awards where part of the funding is to do outreach work. And I think that's very effective. I mean, my lab, we have one of those awards, and we've been to a couple of different public schools in the area talking about the type of work we do. And we cannot talk about our most advanced research because that funding is specifically for youth, for the youth, and to go to schools and talk to young children. But someone could do the same, right? And maybe part of the grant, a small chunk, but some of it should be allocated to, you know, being able to rent a theater or rent a bar for an afternoon once a year and talk about the research that you're doing to the public. In our work, I can tell you it's been a revolution, right? I mean, before introducing a mutation, I mean, again, we work with fruit flies, so they're relatively easy to mutate or to introduce mutations into the genome. But still, it was a relatively cumbersome process that took several months, right? And now with CRISPR, I mean, in a few weeks, you can get an animal that is mutant for a specific protein, or, you know, something really cool that we do in our line of research, not only in fruit flies but also in other animals, is introduce fluorescent proteins next to proteins that we're interested in, that we want to see where they are in cells or how they move in cells. And you can, you know, take the DNA that encodes for that fluorescent protein, and maybe I should explain what fluorescence is. So fluorescent proteins emit light in response to being irradiated with light. So, you know, if you're in the dark and you shine blue light on a sample that has fluorescent proteins, those proteins will emit light that you can detect with a camera. So now you can see where those proteins are. So you can take a fly embryo or a fish embryo, which are transparent, you can put them under the microscope, and if they have those proteins tagged with a fluorescent reporter, you can shine light on them on a microscope, and then they will emit light that you see in the camera, and you can see how different proteins move within cells, right? So that's been awesome because, you know, to do that before we had to introduce the protein already with the fluorescent tag, and what that means is that you have the protein that the animal is making plus the one you have introduced. So now you have doubled or multiplied the dose of that protein, and of course when you add proteins to a system, you can generate phenotypes, meaning you can cause effects in the system. Now you're tagging specifically the protein that the animal is making, so there is no more or less than the animal normally has. It's the protein that the animal has, and you can see what happens when you do that. You can watch the protein. So that's something that we use, I would say, now constantly in our field.